what we're building is, like in philosophy, there's a difference between like changes in degree and changes in kind, right? So it's like, okay, you take a little step, and then you've got like, okay, when you 10x or you 100x something, right? It's like all of a sudden new things get unlocked. Okay, those are like the axioms. I was like, these are the things new. And then I literally went through every manufacturing technique that I could find. I mean, truly, everything down to like, what are they doing with 3D printed glass, at the moment? And you can just kind of like knock these out for a variety of reasons. Our goal is to produce 99% of the world's dynamic biological data. And the reason that that's achievable is because we do not produce that much right now.
Today, my guest is Sterling Hooten. Sterling is the founder of Iku Bio, where he is building a microfluidic bioreactor built on a printed circuit board that cultures, senses, and streams biological data in real time, claiming 10,000x higher experimental throughput at 100x lower cost. It is one of the most niche areas of wet lab automation that I think I've ever discussed on this podcast, and I don't think I would have ever learned about it had I not stumbled across Sterling at an event a few months back, where we had a conversation that was so fascinating that I immediately wished we had filmed it. Sterling, welcome to the podcast.
Thank you for having me. Very big fan. Really enjoy your articles.
Thank you. So I've given a brief introduction of what you're working on at Iku, but I'm sure I oversimplified some things. I'd like to hear your own pitch for what you're doing there and why is it so valuable?
So the largest problems of the 21st century, things in medicine, for climate, for material optimization, all of these are predicated on our ability to manipulate and control living matter. So advancing our understanding of biology is just so fundamental to these problems in the future. And yet the tools that we use right now to interact with biology are primitive. They're primitive in an absolute sense, and they're primitive in a relative sense to what we could be doing. At its core, biology is, time varying, it's parallel, and it's sensitive. And yet the tools that we use right now, that interface destroys at least one of those properties. And in principle, advances in AI also would be an excellent, connection with biology. But that interface is fundamentally broken. So like lab automation right now is stuck at the petri dish and the microtiter plate level. It's equivalent to the like handwriting manuscripts in the 15th century sometimes. And so what we're building is a printing press for biological data. And the way that we're doing that is we're rethinking that interface between compute and biology, and we're replacing traditional microfluidics with a printed circuit board that allows you to embed the fluidics. Cells can live inside of it, and that allows you to communicate and control cells in a way that has not been possible before at high throughput. And the largest application that we see for that is in biologics manufacturing. Right now, biologics, it's a $500 billion industry, and it's supply limited. So every year, Samsung Biologics has to build a new $400 million facility. The reason they're doing that is because you can only get so much out of a traditional a fab, basically a fab plant. They're closer to silicon fabs, actually. And the largest lever that they have is in yield. So how much can you get out of these things? Are they producing? And also, what are the costs? Core of that comes down to literally how many of these dynamic cell culture experiments can you run? And that's a process called media optimization. And it ends up that that core, that one problem, ends up being connected to this $500 billion industry.
It's like if I to paraphrase, if I wanted to increase biologics manufacturing by an order of magnitude, at least my capacity to produce like antibodies and the like, the lever that is most easily pushed on and most like has the likelihood to give you the most like bang for your buck is media optimization.
It is the most bang for your buck. You are unlikely to get 10x on that. The current what you're looking at is how much can I produce per unit time, and then how consistent is that? And those you if you can produce more per unit time, you get higher throughput for the entire kind of facility, and then if you have more stability in the product for biologics and for things that go in our bodies, it's a desirable outcome.
And so my conception of these bioreactors that are producing antibodies is you have a bunch of like CHO cells, maybe sitting in a very large tank. They're sitting in a fluid of media, and they're constantly just excreting out these antibodies that are later then purified. Iku comes in at the step of deciding what media to actually put into this tank. Is that fair to say?
Correct. Yeah.
I've never worked in a wet lab before. My conception of media is that it is sugar water that all cells are generally fine with drinking up. I've learned that this is incorrect, and I'd like to hear your take for what actually is media.
I would say that is a very limited view of what media is. Not incorrect in that if we were talking about media for growing yeast, sugar in water is pretty close to sufficient. But the more powerful way of thinking about media is that it is a very high dimensional control surface for what you can get cells to do, right? Like cellular communication comes through things in the media, right? The media actually is the communication channel in a sense between cells. It's also what carries nutrients into the cells. In mammalian cell culture, it's closer to serum in blood. So it has either many different types of like proteins in it. It'll have different metabolites. It'll have salts. It'll have in defined media, it'll have buffers to keep the pH going. It basically has a lot of components that, and there are hundreds of them really, down to things like magnesium. And each of these are really communicating and interacting with the cells. And they also work across different time periods. So you'll have growth media, which is when you're building up the cells. And then there's media when you really just want them producing these particular things. And right now, if you buy or produce media internally, it tends to be connected to a particular clone or a particular cell line. And so you will optimize the media for that particular cell line, or you'll optimize media for if you're doing, if you're growing neurons. And so it's complicated enough and important enough to the results that you get that exploring it is like very valuable.
Like I know that there are a few companies that have popped up, like claiming to have like redesigned cell lines to make them better at biologics manufacturing. Does that also demand a change in media?
It can demand the key thing is that the biologics that we are producing now are becoming more complicated, and that is making media optimization more difficult. So you do tend to pair the cell line with a media line, both for like repeatability and ease of use, also just for commercial reasons. Like it's a better business. But what really happens is you tend to take a standard growth media or something off the shelf, and then you will kind of customize it for this particular thing that you're trying to make. Because ultimately, productivity is really the interaction of these three things, or three or four things. It's the cell line, it's the media, it's the process conditions, or the tank that you put it in, and then the actual compound of interest and things that you're.
You mentioned earlier about media is both a way, like nutrients for the cell, but is also the substrate upon which they actually communicate with each other. That second part was surprising to me. I did not naturally conceptualize as like cells in a tank actually talking to each other while they're like churning out antibodies. What are they communicating exactly? Like, does that question make sense?
So yeah. I think it's maybe easier to think about it in, the sense of our bodies, right? So, cells will send out, or communicate through different hormones, right? Those will get released. There are small signaling molecules that get kind of like broadcast. Those are carried through the media. In the body, we call it, blood, serum, right? These things. But in the sense, it's media.
Like you mentioned also, you have different stages of media that you want to introduce to the cells depending on the cell's actual life cycle. Is that also true? Is that also true for serum in the human body? Does the body constantly adjust its own sera to whatever the cells need? Okay.
Yeah. I mean, that is the way that cells differentiate.
Is it?
In a way, yeah. Like, you've got some gradient that will happen, and then that gradient which that's basically saying you've got different media, and there's that gradient kind of can tell cells how to orient, or can tell cells how to develop. And from stem cells triggering when what they're going to end up being. That's also basically it becomes media as you add things into the cell environment there.
Mm-hmm.
So
what's stopping me from just replicating human sera for my mammalian cells? Like, isn't that not the best substrate to use?
Well, the first question is, where are you going to get it?
Yeah. Well, like I imagine you could actually, I guess this is a more basic question. Do we understand human sera well enough to perfectly replicate it?
I don't know. What I will say, and that gets closer to what you were talking about originally, is that's what we've been doing historically, but instead of using humans, which not that very limited supply, or limited willing supply, we've been using fetal bovine serum, from cows. There are problems with that. It's highly variable, and for all of biologics manufacturing, the goal is reduce variability. And if one of your largest inputs is variable, that's a problem. It's also a challenge because things like you can't sterilize it in the traditional way. You can filter it, but you can't heat it up without destroying. And things like prions, which could be quite bad, you'd need to prevent those coming in. So the industry has really moved much towards formulated medias. So you're building it up from the constituent parts, and that also allows you to be, it reduces variation and gives you a lot more control than over how are you particularly tuning that media.
When you say at some point fetal bovine serum was being used,
it's still in use.
Oh,
it's mainly in use in research. I think I'm maybe there are some biologics manufacturers who are using fetal bovine serum. I don't know, but I think the industry has pretty much moved to.
At this point, would you consider that the quote unquote synthetic serums that are attempting to recapitulate the biochemical properties of fetal bovine serum, the synthetic stuff is better? Or is it just it's easier to get So you're okay with not perfectly capturing fetal bovine serum?
I think it's better.
Okay.
I think it's better. And I think it's better in that you, again, get to tune it.
Yeah.
You get to tune it.
Okay. And so attempting to be more concrete about what is a media optimization engineer exactly doing? Let's say I have a plate of CHO cells. I want to produce Keytruda, so Pembro. I have a bunch of cells. I have all of them are willing to produce the drug. They've been genetically edited to do that. What's the next step?
So the process in general is guess and check. So you will take a cell line that you've edited or produced for this. Most of the time, it's just, and then you'll take it out from the freezer. You're gonna grow it up a little bit, and then you'll probably take 4 or 5 of those because you don't kind of know yet, right? Like which particular strain will do best.
So you're trying with multiple strains.
You're going to try with multiple strains, and then you will run experiments that allow you to. First, you're going to run it in microtiter plates normally, right? And you're going to just see kind of where are we. Where does it look? Which of these cell lines kind of seems like it fits best with these? After you've kind of narrowed it down, you're going to move to something that has more control. And the reason that you're going to move to something that has more control is that what happens in a microtiter plate is extremely disconnected from what happens in any kind of production environment. And the core reason for that has to do with flow. So in a microtiter plate, you get a lot of sort of like capillary issues, right? It changes you've got the surface tension kind of comes up. That changes the gas exchange rates. You get evaporation, and you don't get any of the different kind of like gradients or different little bits of shear forces, all of these things that actually affect how cells grow in a large reactor. So what you do is you put it into what's called a benchtop bioreactor. And so this is, a little bit bigger than a Fiji bottle in terms of what it'll contain. And it's got an impeller in there and it'll spin it around. So now you're going to grow those cells in that media for 10 days or something, right? And during that time, you're going to also change or control the pH level that's in there. You're going to control the temperature. You're going to, you'll set different, impeller rates, kind of seeing what's optimal, and you're going to run that for. One person can maybe run 12 of those experiments, 15 of those experiments. It's pretty laborious right now to actually set those up. It's going to run, and during that time, you're going to pull off some samples. You'll take those to the analytics section, depending on how booked up that is. That could be 3 days to a week sometimes to get all of your answers there, and then so you'll do that.
And sorry, what questions are you asking at that point? Like, what are the sample meant to answer?
Right. So ultimately, your sample is meant to answer how much total biologic did we produce in here, at what quality, right? And then the other question there is like, how overall, like how consistent is it? Will it be? That's actually a large sort of hidden cost. Is, as I said, like the best way to think about biologics manufacturing is to think about it as high precision manufacturing, and closer to semiconductor manufacturing. That's really the reason why like Samsung Biologics is in the position that they are. It's because they took what they learned in terms of process control, they brought that over. the reason that Fujifilm is a large manufacturer is because they took chemical process engineering and they brought it over. Now, these were not biological companies, right? They are industrial manufacturing companies. And when you think about reducing process variability, one way of looking at that is just how precise is the part that comes out, kind of. But then what makes up that, right, is like, basically how much variation can we absorb without it affecting the end product. And so if you can come up with media and process conditions that are kind of more forgiving, you're relaxing it a bit, right? It's like you can still end up with something that's very precise at the end, but we didn't actually need as much. We were kind of more forgiving over here, and that can be important because if you lose a batch of biologics, it's very expensive, and that can happen, and it does happen. And so the way to reduce that is through there's media optimization. And so to finish on this, you've run that set of experiments. You've got your readout there, and those readouts, although those are like the most important, you're also going to characterize kind of everything in there that you can, because you want to see how those are affecting that actual readout. Then you will repeat this, and depending on how much time you have, maybe you will get 3 or 4 kind of runs at that, and then that's it. And that comes down for biologics manufacturing to the regulatory reasons.
How much of what you decide are like the optimal cell lines and the optimal media? It's like there is like a threshold of quality you want to meet, and after that you're done, versus you're trying to make this as like perfect as possible. Was it kind of dependent on what drug you're trying to produce?
I think the goal is match what was in the phase 3 trials.
Okay.
So in the process of taking a drug to market, during your phase 3 trials, the batch that you produced there, that is what all of the FDA's kind of evaluation was based on. So they want to keep that as the same. So anything that deviates from that is undesirable.
Is this true even when the drug gets to the generics? Like it gets off patent, and do the generics manufacturers, are they trying to make it even? They're trying to improve the process even more, or even for them, they're trying to replicate exactly what went on for the original, or with the original company?
That is a great question. I should look into that, because they do have to go through them. So they have a couple options. The first thing is that they will basically just license the cell and the media from the existing pharma company, right? Pay them for that, and then that way the pharma company can still get some revenue from that. The alternative is they need to come up with their own cell and do it. I think the regulations are such that there's a way of, I think it's like they can prove, if you can prove that it's similar enough at all, then it just counts as like a process change.
Mm-hmm. And like getting back to the question of actual media optimization, like the media optimization person goes to the analytical chemist. The chemist tells you like all you need to know about the samples that you've been given. You repeat this 6 to 5 times. What are all like the levers of change that you have over the media?
So media is best thought of as this control surface for affecting what the cells are doing. It's like, what are the levers in there? you can change the components, and then you can change concentration of those components, and then you can change timing of those things. And if you start with 200 or more, but let's start with 200 components that you can put in there, and then the different concentrations that they come in, and then the timing, that already is like quite a large space to explore. Then you have that interacting with the cell and the different cell lines, larger space, and then with that fixed compound that you're looking for. So the standard things that people are going to change or tune in there, right, is when is a carbon source coming in, and when what as you start producing different proteins, the need of the cell change. So if you shift into a different mode for the cell, you can kind of signal it to shift into a different mode. Starts producing these other, all of a sudden its needs change, and being able to anticipate, buffer, and meet those needs, that then has a lot to do with the output.
How much of the optimization, like even the direction or specifics of the optimization, can be theoretically known and applied versus just always empirically determined? I guess the more specific question I'm asking is does a media optimization engineer, are they coming to every new problem almost like tabula rasa? They have like whatever experience they had in the past does not apply to this new cell line with this new drug.
So the question of how tractable is this of a problem, and what's the current state of the art? The current state of the art is that things, best practices live in the mind of the practitioners, and a lot of that comes down to familiarity with that cell line, familiarity with the media they already have, and most manufacturers are working in a particular kind of domain or specialty, right? And so as you're constraining that search space, it does make it easier to operate in there. However, it is not the case that you will one-shot it coming through. And then the second thing is it's actually reasonably easy to get caught in a local maxima. And if you don't, if the cost of running those experiments or experiments themselves are sort of precious, you're really not going to push very far out. The lever they currently use is mainly in strain engineering, and so they'll try to select strains that'll have the highest performance. But once those are set, or those cells that you're using right there, it does all come down to the media. For optimization, in a model sense, it does seem that it's tractable. It does seem like that there's transfer learning. How broad of that really comes down to what experiments have we been able to feed things so far into these models, and the answer is not very many. Like the largest facility that I know of for running sort of dynamic cell culture experiments, they can run 300
in parallel at any given time.
300, and that's the entire company is just doing that. So that's the state of the art, or that's kind of where it is, and a lot of that comes back to the fact that it's so manual.
Mm-hmm. The one last question I have before we move on to how Iku is fixing this is, I can understand being able to easily modify concentration of the media.
Mm-hmm.
I understand being able to modify the timing of when you're giving which media to the cell line. The the components, the constituent components, feels a lot more complicated because that's like 200 components. How much of that is like, in practice, there's like 10 of them you modify at any given time, and the other 190 are like pretty standard, and all cell lines will need this.
Yeah. So how much is like what's the core? Is there some is there
like
some
an 80
dimensionality reduction?
Yeah, like is there an
that you can do?
80-20 thing going
Yeah.
on? Oh yeah, yeah.
Absolutely.
Okay.
Absolutely. Which as I said, like the glucose, your sugar source or carbon source, energy, the the pH that you're running at, those are there probably are 10 that are dominating it, but that's why it's actually why it's so challenging is because there are 10 that are dominating, but because the system that we're controlling is quite nonlinear, it can amplify what are sometimes, in certain conditions, some small change. And my favorite example of this is that literally just like change. It turned out this was in industrial manufacturing, but like changing the amount of magnesium at a particular point, like doubled the output. And it didn't necessarily need, there was no like a priori way of knowing like, oh, it would have been magnesium that like went in here. And you say, oh, okay, well, sure, like that's a lever and we should do that on each of these. But the problem is that potential exists for all of those other 190 things or all those other things, right? So it's like, sure, there are these core things that tend to like dominate,
But those 10 things could vary based on what the problem actually is.
Yeah. Okay. Well, those like the core things of like you do need to, the salts that are in there, right? And the and what when energy comes into the system, those are definitely floor level. You have to figure those out for it. Okay, but then and if you get those wrong, basically like those are controlling kind of the where the floor is. So if you get those wrong, it kind of doesn't matter what you do in these other areas. You're not going to have high performance. Mm-hmm. But just because you get those right doesn't mean that you have high performance at all. It's like they're just like those are table stakes. You need to get those done.
That makes sense. And so like we mentioned this engineer who's trying to produce Keytruda.
Sure.
They're evidently building like at the very beginning building in a Fiji shaped bioreactor.
Yep.
Doing these rounds of iteration, trying to get to something good. What is Iku's proposal for what is a better way to do it?
Our proposal is to rethink what it is that you're trying to do when you run that experiment. So that Fiji bottle device gets used for two kind of purposes. One of which is like you want to grow cells and you want to grow them to feed a seed train. So you're growing them, or you need like that quantity of those cells. That's one. And the second is that you need information and you need to be able to control the environment that the cells are in over time in order to get it. And so for this first set of things where you're trying to grow a lot of cells or grow them up, great, perfect use for it. If you're trying to extract the most amount of information and try to control the cells, it's a very limited way of doing it. Before starting on any of this, I'd actually seen some of these benchtop reactors, and I asked them like, if the thesis is that it gets better when you go smaller, why did you stop at the Fiji bottle? And the answer was, well, if we go any smaller, our sensors won't fit. And that's because they're using off-the-shelf sensors. And if you ever see a photo of these things, it's a hodgepodge of different things that have been kind of like crammed in there. And that literally is like, they doing sensor design is its own field. And you need to design not just one type of sensor, you need to design many different types of sensors. And there's also not that much of a benefit going from, a Fiji bottle to half a Fiji bottle in size because of the manual labor and all these things. So our solution is to think about what's actually the best platform for building sensors. And then can you put cells inside of it? And my last company was a robotics company. We, any of the humanoids now that you see going on, I'm highly skeptical of the economics on these things. But any of the humanoids that you're going on, the core technology that enables them to like move, interact with the environment, that was what we built. And that is a sensor problem, and it's a sensor in a high noise environment. And that is abstractly quite close to what we're doing in biology, right? Mm-hmm. The idea is if you have a good place for building and placing sensors of different types around, now you've reduced the problem. And so easy place to build sensors. Now you just have to figure out how to grow cells inside of it and keep them alive. And if you pick a mass manufacturable technique for doing that, it also solves some of the scaling problems, because the challenge with controllable systems right now is that they still literally require somebody to come over, unhook everything, set it up. You can use disposables to sort of take that down a bit, but it also takes, when you go larger, it takes more media. It's more expensive to run it. It's less repeatable. None of it makes sense except for it's a difficult engineering problem.
In like a practical sense, like I can buy that. Like this form factor was chosen purely because our sensors are not small enough to fit in something smaller. What is the form factor that you guys have?
So the core differentiator is that we are reusing printed circuit boards, which are ubiquitous. They are in your phone, in your microwave. And we put microfluidic channels inside of them. And by doing that, it allows you to then have cells live inside. They can pass through, they can live inside there. And it turns out that making microfluidics previously that integrate those types of sensors is extremely awkward. And so you either don't do it, or if you do it, it's still hand finished. And so the big differentiator is everything comes straight from the fabricator, ready to go. And this is a theme that has happened before. So in like silicon photonics, which is where you take existing silicon fabs and you say, hey, can we use this in a new way? And not to just do like integrated circuits, but can we now do things with light in it? In your iPhone, the has like a LiDAR detector. That was like a new way of using that. And the core there is that the process that's used is called lithography, which is where you'll take a mass kind of like a snowflake, you project light down through that or something, and that cause certain things to react and certain things not. And lithography is a really powerful manufacturing technique because you get complexity for free. What that means is normally if you're doing like traditional subtractive manufacturing, as your part gets like more complex, you've got more nooks and crannies in here. It takes more time to make it. Or you've got more tool changes or all these things. But with lithography, you pay that cost once. You pay that cost when you make your snowflake. But it actually doesn't matter how complicated you make the snowflake for what's down here. And so it pushes you to say like, what's the most complicated thing we can make here that has the most value? because it literally costs the same. It doesn't matter if it's one line through here or some complicated maze. So that's what semiconductors are doing. Then they apply that to photonics, right? LiDAR. Printed circuit boards are made the same way. It's lithography, and if you can leverage that in more complicated ways, you start both enabling capabilities that weren't possible before, and also are writing like a cost curve that's really beneficial. So the idea is like every time that we have found as a society a new use for lithography, like large industries get built off of that.
And sorry, so where's the lithography component coming in when you're talking about like building a new bioreactor?
So the way that we make our chips,
which you have,
right? Yeah. Let's do we?
Oh man,
Here.
It comes out pretty small. Okay,
you hold it. Yeah.
Okay, so I am seeing that there's a bunch of like circuits coming out from here. Where like walk me through the anatomy of this device.
So the first thing is that it looks kind of cohesive, but it's actually 6 layers, and each layer either is carrying electrical signals or fluidics, or routing fluidics in there. And so for this particular chip, it has a channel that's a millimeter wide and about the size of a human hair tall, and that's actually like a great size for cells. And you can flow media cells into it, and then it has all of the components that sort of like a benchtop bioreactor or a more controllable system would have. And the way that you make these is through lithography. So these lines and all of the features that are on here there's a snowflake kind of pattern that's made for that, and then they will put what's called a resist and an etch on, and so it will kind of keep those lines where you want them and etch away everything else, and then you make the next layer, and then you make the next layer, and then you compress all of those together. And so the way to think about it is it's like a 2.5D space. So you've got two-dimensional plane, but you've got multiple two-dimensional planes. And so topologically, that's going to allow you to like do things like take a spiral, get to the middle, and you need to get out of it. And so you can come up and out and away and round. And it also allows you to put electrodes or different sensors in relation to the fluid, in relation to the cells in different places. And that's kind of abstract, but let me give you a very concrete example, which would be if you want to have a readout of electrical signals of like heart cells, cardiomyocytes, you want to read like across those cells. Well, you need to be able to put you need to be able to put electrodes above and below them normally, right? Or you can put them side to side, right? If you're trying to do these things. That's like a primitive. Like that is it sounds very simple. And yet, I will tell you, that is, with other techniques, a difficult thing to do. And so by switching to this new substrate, a whole class of problems that are traditionally quite difficult become substantially easier.
And sorry, I don't have a great conception of like where do the cells like like on this like green thing, like are those like holes like where you put in the cells?
It is really and actually I have a drawing I should send you. You can put up a drawing on this picture. Because that is also part of the problem, is that from the outside it literally looks the same as any printed circuit board. Second thing is, in biotech, a printed circuit board looks like alien technology. Okay. But yeah, it has actually small holes. There are ways of getting fluids into the actual device, and then you can run them past sensors, or you can run. It's often easier to run the fluid past the cells, and then you're kind of reading things out on the fluid.
And so there's not like a specific chamber here where the cells sit. They are literally like in a line formation as you run fluids through them.
In this particular chip, this particular chip is like a year old. In newer designs, you have more like a chamber.
Okay.
And you're kind of seed that chamber, and then your cells are growing over it. But the powerful thing about using this technique for making microfluidics is that you can make a large number of variations, and that's a difficult problem in traditional microfluidics because you would need to make new molds, and a new mold is $25,000 or something, $40,000. You need to get a mold maker to come in and machine it. Your economics on that mean that you need to make a lot of them. With printed circuit boards, it's easier to make variations to them and then just do that. So we have kind of a core catalog that we're building of these are the designs for particular applications. But every new printing, it's relatively easy to change it to whatever the condition is.
Sorry, is it fair to say that typically microfluidics are not built using lithography, but you are building them with lithography?
Microfluidics historically started with lithography. So they started with lithography. They were built using similar techniques in the use for semiconductors, and in most research labs, when people build microfluidics, that's still the way it's done. Okay. What you'll do is you will make a silicon mold, and then you cast a polymer over it. This polymer is called PDMS, and the desirable properties of it is that it's optically not transparent, but you can at least see into it, and it's gas permeable, and so that allows you to have exchange of gases without. It's like you can put it in an incubator and you can use it there. Downside of that is you can also get evaporation. The problems with that is you end up with a kind of a fragile output, and it's also still fairly labor intensive to do that. But people like it because you can do it, in your own lab. The difference comes down to the use of lithography for the sensors and fluidic channels together in this thing, and critically for silicon fabs, you need to be really careful about contaminants. So if you need, for example, like a gold-plated electrode, you cannot do that in a silicon fab because you will contaminate the fab. It's like not allowed at all. Very bad. So with the printed circuit board as a medium, basically, you can integrate many more different types of sensor modalities than are sort of possible with silicon. And then the second thing is just on a like the reason to use silicon is because you want extremely fine features and detail. Once you need something on the nanometer scale, it's kind of the only option. But our thesis is that cells themselves are more on the 5 micron scale, which is few orders of magnitude difference.
Yeah.
And that's actually the domain where printed circuit boards are a better place.
Is there so if historically people do use lithography for microfluidics,
Mm-hmm.
but they only use it for the channels and not the actual electronics, what what innovation allowed you to actually include electronics in the design of the microfluidic?
Yeah. So let me state that so microfluidics is like a really broad term. For example, DNA sequencing, Illumina, right? Like that's using silicon for for a microfluidic system
Mm-hmm.
and doing sensors. It's a really useful place for doing that, but it has limitations in terms of where in space you can place things. The example I gave earlier about trying to read across these cardiomyocytes, you can't do that with silicon. There's no kind of way to build a channel that size that you need for the cells to go through it, but it's buried and you have electrodes above and it's just like you just can't make it that way. So the core innovation is first of all just conceptually thinking about printed circuit boards as a media for making as like a medium for making microfluidics.
Mm-hmm.
I'd been working with circuit boards for 10 years or something. Never occurred to me to put fluidics into them. Been talking to people about this for 3 years. Never met anybody who was like, "Oh yeah, like I've seen that before."
So, like as of today, there's no one combining circuit boards with microfluidics.
There is for diagnostics.
Oh, okay.
Yeah. So the professor Despina Mashu at the University of Bath, she's really like the pioneer of putting fluids into the circuit board from the fabricator. And the reason like that's so important that I keep coming back to it is you can do a lot of things, and academics are prone to this. You can do a lot of things by hand. That does not scale if you need to make hundreds of thousands or a million of things, right? If you're doing that, you need to pick something that is mass manufacturable. So in terms of, cost and complexity, the cheapest thing to mass manufacture for microfluidics is either like paper or molded things when you build a lot of it. But if you try to make microfluidics in a PCB in a lab, Jerome, you can do all kinds of weird things. Getting it so that it's compatible with the standard fabrication process, that's a different ask. Both because they're not terribly keen on changing their processes for the most part. But then the second thing is that when you do it by hand, you're introducing variability from the beginning. When you have it done in a fabricator, you're inheriting the hundreds of billions of dollars that have been spent cumulatively on like printed circuit board development. Like it's been around for 60 years. Like entire industries are built upon it being already very good. So let's just reuse that thing that's already quite good and low variability.
Could you give me some intuition as to like how the device is actually put together? So, okay, like my mental conception of lithography is like, yeah, you're able to create these like very fine channels in the silicon via like shining light through a mask. What's the next step after that? Like maybe you do this on multiple layers. Do you have like this multi-layered system of just like channeled almost?
Yeah. So for traditional silicon fabrication, it really is like it's a mask and then you etch, and then it's a mask and you etch, and mask and you etch. With printed circuit boards, it's more like each layer can be made out of different materials. So this is where like there's an enormous amount of flexibility in terms of it's a much richer palette to to start building out of. So the foundation is what's called FR4, which is a fiberglass structure. That's why they're normally green. It's this fiberglass structure, and on top of it'll come coated in a layer of copper, on the bottom, and that is the simplest circuit board that you will buy. The cheapest one is just that, and it's just been etched, and then they will put down what's called like basically a protective layer on it. So that you don't kind of just like scratch off the copper, and then you'll silk screen it, which is if you want to put labelings and all these things. But that at its core, like that's what the process is. When you add in microfluidics, there are techniques for being able to make the fluidic channels on one layer, and then as you need, you can just kind of like stack on another layer, and then that layer has fluidics. Or in between them, now you can route like your heaters, right? You need to put your heaters there. Or if you want to put the electrodes or whatever your kind of like end sensor is, you'll pattern that on that layer, and then you sort of build it up, and then you kind of stack them together, you close it, and then.
So like in V2 of this device, you have this chamber where the cells live.
Mm-hmm.
You have microfluidics connecting this internal chamber, maybe it's external, to like a bunch of pipes that like feed in like some particular access or variation that you want to control during the media optimization process. And you also have like embedded or maybe external sensors that are connected to the circuit board to have some sort of readout of what's going on in this chamber where the cells live as the media is being applied, and like what's the output. What is the output of the system? I imagine like one is maybe temperature, maybe another is internal humidity. What other axes are there that you can actually get straight off the sensor and straight off the device?
So the way to think about it is that if you're going to do any kind of cell culture, there are a set of table stakes that you sort of need to be able to do in there, and those are temperature, pH, and dissolved oxygen. We're flowing things through, so you need to be able to measure flow rate. Mm-hmm. And those together, that's the core set of things that our system is currently like reading from. The next layer are the like electrochemical sensors. So being able to read impedance is actually very useful. If you can read impedance for the media itself, you can detect some changes in how the media is adopting. And if you place them in relation to the cells, you can also correlate cell growth with impedance, which is based on the kind of like based on how these charges sort of end up hitting against cell walls at different frequencies. So like that's like a core thing there. You can do conductivity through it, which is partially used for offsetting where the impedance reading is coming from, because the impedance can sort of get it can get interfered with in a lot of ways, and so you sort of like need a reference point in order to do that. And then you can do other electrochemical techniques like cyclic voltammetry. But the readouts right now are the impedance, the flow, dissolved oxygen, pH, and temperature.
Theoretically, I imagine like all of these sensors already had miniaturized versions of them available. Is that true? Not true?
Not the case.
Not the case.
Not the case. There are nothing that our system can do at the moment is anything that you couldn't have done by hand or with a very custom setup. The challenge is, how do you do more than 2 of those, 3 of those at a time? How do you build them, economically? For example, like the chip that I showed you, in any kind of reasonable quantities, it's like $4 or something, $3, and that's actually still even. You can get it down to like less than $1 on that. So if you're buying sensors off the shelf, that's not, the economics are going to start killing you very quickly. And then the second thing is, it's a challenge to integrate those things. So, a big idea in robotics or engineering any kind of real system is that interfaces and connectors are what will kill you. They're very common points of failure. So the best solution is like no connectors. When you build sensors all in the same platform, you essentially get to do it with no connectors. So that's the trade-off is more difficult engineering from the outset, but lower variability and better economics at the outset.
I imagine you get like dissolved oxygen, pH, and a few of these other parameters. I imagine like there's still some you were missing, in the sense of like is the protein that I'm expecting to be produced is actually being produced?
Yeah.
So like it sounds like you're cutting. You're allowed to optimize to a threshold, and then after that, you need the analytical chemist to come back in and like do their thing.
So our goal is to make the analytical chemist kind of a confirmation rather than be limited by it. And the reason comes down to sort of lessons from control theory. So the first is that any system that you're trying to control, in this case cells, if they move at a certain rate or certain speed, and you want to be able to dampen that or amplify it, right? You need to be able to read it fast enough that you can come in and make an intervention. Anytime that you take a sample of something and do an offline measurement, that loop is normally too long, right? Sometimes that loop is 5 minutes or 2 minutes. Okay, maybe you can work with that. If you need to take something to your analytical chemist, it's probably hours or days. That information is not useful to you in the actual control of the culture, right? So what you want are real-time sensors. You want sensors that are truly like integrated into the thing. For the sensors that we're using now, that really is just the table stakes to enable us to start building in these other sensors. If you don't have those core sensors, you can't even keep the cells alive. You can't, there's just no point. But things being able to have live readouts of like monoclonal antibodies, that is what we're building towards in the device. It's being able to have the optical sensors built in. It's being able to leverage the sort of biological techniques or chemical biology techniques that we have right now for getting signals out of cells. All of those are like compatible with our system, and that's where I think like the real value starts becoming unlocked, because there's a large difference between, like sort of philosophically, between just reducing the cost of something versus what questions become askable now. And the questions that become askable and sort of like experiments that you could run. That's what I think is so powerful about using this substrate as a technique is like, okay, you make this core thing. Can you grow cells like in high throughput in this dynamic way? Okay, once you have that, every new sensor system you put in gives you more lenses into it. And this comes back to why lithography is so powerful, is that normally you have to make a trade-off, right? Normally, you're like, every sensor I put in costs me money. And so I'm only going to put in the sensors that I need here. But if it doesn't cost us anymore, or if it's basically trivial, then the idea is actually, let's just instrument it. Let's just keep instrumenting it. And classically, you would say like, well, I don't really care about those features and those things. Those things don't matter. But what we're moving towards is more of of having fewer priors and having less human interpretation on the streams of data that are coming in. And so, for example, like the impedance sensing does not give you a simple number that comes out. It's a complex number that comes out. Okay, whatever, you could still deal with that. But then it's a complex number across hundreds of frequencies. So you're getting back this large readout. All right. And then it's changing over time. So if you and I try to decode that, it can be difficult, right? And we can argue about this. But, machine learning is getting pretty good, arguably quite good at handling those types of things. And so the way that I separate these two, the way we're separating these two is they're what are called narrowband sensors, and then they're broadband sensors. So a narrowband sensor is, for example, like readout on temperature. You're going to resolve that to some either resistor variable or some temp or some on some basis like Celsius, and you want that to pretty much just respond to temperature, not respond to anything else. Right? Okay, very easy thing to interpret. Same way with your lactate. That's the, you want something that only responds to the lactate in the media, nothing coming out. These are narrow band sensors. They're meant to reject kind of everything else. And then there are what I'm going to call these wider band sensors, which is, if you take a microscope and put it on something, that's a fairly wide band, right? It's like there's a lot of stuff going on in there. There's not kind of just like one answer about what's going on. And you can sort of select like, oh, I think these things are more relevant to the questions I'm asking or not. And things like optical, the impedance, some of these other electrochemical techniques, the magnetic fields that are in there. When you have machine learning on the other end to interpret that, it would be surprising to me that you would not, that's not useful.
This is maybe a naive question, but at the end of the day, all the signal you're able to extract from like this device is going to be some like electrical property of the cell, of like the tiny little bioreactor you have in there. Is that correct?
No, we're the big picture is that we're integrating all of these different modalities. So we are integrating the optical modality. We are, my dream here is to get like Raman sensing into multiplexing Raman sensing across this, right? For having that method of looking at it. It's having those with the lactate and the glucose and the your monoclonal antibody sort of like readout, right? Or whatever those domains are in an instrument sense, that's extremely powerful. So that's the goal.
Okay, interesting. The I imagine some of these variables, like you mentioned, are like immediately interpretable. There's like a good value you should be reaching. Imagine like dissolved oxygen is one of those. For the more complicated ones where it's like you don't know whether this is a good value or a bad value, like glucose or
Mhm.
some other mineral, where does like the ground truth come in? Is that where the analytical chemist comes in and like they give one singular data point? It's like what's good, and then the purpose of the system is to like correlate everything that you put into the system and like all these output variables
Right.
you got out to that ground truth or something else.
So I think a useful lens for this is what's his name? Richard Hubbings is from a book called How to Measure Anything. Highly recommend this book. Book changed my life. And the idea is the expected value of perfect information, and it's that any reduction in uncertainty has some cost to it. And so when we're taking a measurement, there's an economic aspect to that, and therefore a trade-off. So, knowing the temperature of this room, there's not much value to us, right? It doesn't matter whether we're off 5 degrees, or 0.1 degrees. For again, semiconductor manufacturing matters quite a lot, right? You need really really tight value there. So if you take that lens and you say, certainly like overall, there's a need to have precision on the readouts of like how much antibody do we get out of this, and the quality of that, right? But earlier parts of the process, like, do you need that level of precision?
Well, I guess like at the end of the day, I imagine like the whole purpose of the process is to get to antibody production. But I guess like, or is like part of what you're saying that there are earlier intermediate benchmarks you want to hit before you get to the antibody. Okay.
What I'm saying is that your ultimate readout, right, is like yield, titer, quality, and stability over these things. Those are the things you care about, and pretty much in that order. Okay. Even on the yield, though, you're still going to get, there's still variation inherent in cells, right?
Mm.
Every batch you run, even though they're trying to reduce variability, you're still going to get some variation in there. So if you take a sample and you learn to 2 decimal points, the titer that came out of that, the yield that came out of that, okay, great. But like your process variability is like 1% anyway, or something, 2% anyway. So knowing it to 3 decimal places doesn't really help you. And then the second part of it is like, if you can cut down on the number of, like, if every measurement has a cost in some sense, can you change your measurement system such that you get the information that you need in a more economical way? And part of the way of doing that is by loosening constraints when possible. So ultimately, certainly, you still need you still are going to run, you're still going to run it on your benchtop and your pilot things, and you are going to characterize it there, right? Because you do need like ground truth from those things. But in terms of which is the right media or conditions to get to in there, okay, do you need 2 decimal points of accuracy on that? Do you need all of those readouts to do it? No.
Is a good way of thinking about this is like you start with the EQ device at the very beginning, and then once you're happy with what you see, then you move on to the benchtop device, allowing you to like narrow your search space down to like a very small number of parameters.
Right. It would basically be like you're still going to end up, the process looks pretty much the same. The difference is, what is the quality and speed that you came to that. What's the quality of the answer you came to? What's the speed that you came to it? And then the second part is, how many of those benchtop experiments did you need to run? Because there's a difference between running them in an exploratory sense versus running them in a validation sense, right? In a validation sense, you're just trying to make sure that things are like repeatable. So you need to run, let's say 3 to 5, copies of it or something. But if you're already quite confident that you're that you're at the optimal point, it's like doesn't make sense to do that sort of experimentation there anymore.
Yeah. Moving on to okay, you've done the eco optimization, and now it's time to move on to the bigger things. How worried are you that moving the cells to a physically larger space forces the media optimization to move in a completely different direction?
It's definitely possible, and every time that you change physical, shape and geometry, right, you do get some variation there. The confidence comes from understanding that, first of all, empirically, every microfluidic system that has flow integrated into it ends up correlating quite well with large systems. The reason that people have hesitation about it is because they think about microfluidics that doesn't have flow and the recirculation effects. And that's actually the key thing, right? It's a question of do you have flow, this thing or not? And how does that flow and those shear forces and the oxygen, transfer rates and the gradients that you create, right? How are those representative at all of what's going on here? So that's one part of it. But let's say you don't buy any of that. The easier way is that it actually decomposes into two broad parts. There are parameters that change with scale. So these are things like your hydrostatic pressure definitely changes with scale, right? You're not getting away from that. Certain mixing times, these change. You can get pockets in very large reactors, right? These change. But then there are a set of parameters that empirically don't seem to be scale invariant. And for the most part, media optimization seems to be scale invariant.
Do you imagine, in the ideal setting, this is a closed loop system that just continuously tries different media optimization parameters, feeds it all into a model, it plans the next round of media optimization, that just goes in a loop?
Yeah. So how does aside from running the experiments, how do you actually interpret and decide with it? So clearly the entire zeitgeist right now is about replacing the control layer with AI and models. And whether you can do that on experimental design from reading a bunch of papers and then this is the thing I'm going to build, I'm less convinced that that's necessarily the best way. But for these types of experiments, certainly seems the way. It's actually key for making the whole product, because otherwise you're handed so much information back that the problem then shifts to processing it. So one of the lessons that I've taken from talking to people who have tried things in media optimization, tried doing cloud labs or doing these things, is there's a lot of hesitation around sharing cell lines. Understandable. And it also comes down to information about what the what the result of those cell lines are. So for example, a company that was running experiments externally was not allowed to look at the results of some of these analyses. I was in their contract that they're not allowed to actually look at the results. Okay. So it's really hard to improve or build your own model if you cannot look at the results. What we're building is a federated model that allows the customers on site to run the device. They can pull the model get a new experiment design that runs in there, and then the model weights are updated, right? This is the same way that the Tesla, self-driving was trained, right? Federated learning, resolves that IP sharing complaint or constraint. And the reason that's so powerful is that now you have a model that is learning from diverse experiments across different cell lines at different places, but still on the same hardware. And that's really key because otherwise there's too much experimental variability in the data you're getting back, and so you're not going to generalize well on that. And the sort of hedged bet here is that if it's not tractable through machine learning and models, we are still building the highest throughput, most economic way and fastest way to get to that answer through still running experiments. And if it is tractable, we're going to have the best model for running those experiments. And I think the answer is actually going to be a blend of both. I do not believe that experimentation is going away, but I do think that we will be able to get to much better answers much faster, because that's really the ideal, right? Like the ideal is you can then once you have that model, now you can feed it in even earlier in the process, right? When you're doing your strain engineering. So coupling those together becomes possible once you have a model.
What parameters does the model actually intake into? I imagine it'll take in all the inputs you give into the system,
mm-hmm.
all the outputs you get out of the
mm-hmm.
system, and maybe what the system is actually meant to produce and the strain itself. Mm-hmm. Is that everything, or are there others?
That's. I think that's a complete.
Okay.
I think that's a complete view.
If the belief is that you'll probably still need human experimentation to help the system along, and maybe the ML won't fix everything zero shot. Can I conceptualize this as like there are 10x media optimization engineers, and they'll be able to iterate much faster on this model system as a result of that? Or do you imagine media, bioprocess engineering, is a pretty standardized field where these are the first 10,000 things you try, and maybe in the old world you get to try 5% of that, and in the new world you try the 1,000 things, but ultimately it's the same set of parameters that you're tuning that the media optimization engineer is tuning.
Yeah. So are we tuning a different, a larger set of things rather than just the engineer?
Yeah. I guess specifically all the knobs that the engineer usually gets to tune,
mm-hmm.
do they also get to tune in the system, or is it a subset, or maybe even larger?
It's a superset.
Superset. Okay.
It's a superset. You're getting to tune far more. And it's a superset in a few different senses. The first is that just bringing the economics down, making it automatic, allows you to even if you had the capability previously to change a variable, you wouldn't be able to you wouldn't essentially have the budget or the time budget or the capital budget, right, to actually exploit it. That's one sense. The second is that it allows you to make finer interventions, and with more feedback built in. So the reason for having the real-time sensors, why it's important, is what you actually want to do is be able to anticipate what the cell wants before it needs it. Because there's always a delay between when something gets introduced into the environment to when it gets uptaken by the cell, right? So ideally, actually, you want to see those signals happening before the cell needs it. Now, in order to do that, you need real-time sensors that are kind of picking up on that and starting to match that. So that's a domain that's just not kind of possible.
Yeah.
in other systems. So.
I'm curious about. I assume there are microfluidic bioreactor systems that at least exist in the literature. How much improvement do people generally see by going for these systems versus like the Fiji shaped benchtop?
Right now, I would say close to zero.
Okay.
And the reason is economic. So the one metric or lens for looking at it is just what is the sort of cost, all in cost to getting that dynamic cell culture data, that one experiment, that data. And there's two components to that. The first is like, what's your CapEx, right? Like how much did it cost to actually get this device in here and use this thing? And what's like, and it's really this like CapEx per experimental lane. And then the second is, what is the OpEx on that? Like every time that we run the experiment, how much does that cost? And so to give an example, like the benchtop reactors, depending on whether you're going with kind of the gold standard or some of the derivative ones now, let's say the CapEx is between $5,000 to $15,000, $20,000 for each experimental lane, and then your OpEx is, all because you've got not just the media, you need to also take the time to grow the cells up to be able to seed it. You've got the human coming in and running it, and then you've got the actual disposable, or you've got cleaning the thing and sterilizing it. So it ends up being around $1,500, $2,000, right? Every experiment that you run. The closest like microfluidic system in capability, it's only 4 lanes, and it's $80,000. And so that gives you a per lane cost of still $20,000. And then the disposable costs are I think still around, $500, $700 for each thing. So it's just like there's not much economic reason to do it. The reason that product is on the market is because it cuts down on media utilization. But that's why I think it's not a very successful product. What we're building is, like in philosophy, there's a difference between like changes in degree and changes in kind, right? So it's like, okay, you take a little step, and it's just like, okay, it's different, but it's not like qualitatively that different. And then you've got like, okay, when you 10x or you 100x something, right? It's like all of a sudden new things get unlocked. And so we're looking at, a CapEx of $8 a lane. And we're looking at an OpEx per experiment of like $20 or less, right? And so those two things together really transform what it is. And then if as I said, you start integrating more sensor systems into it, those two parts are kind of fixed, right? The CapEx and mostly OpEx on that. But the amount of data and the amount of value that you can get out of it, that's where I think like there's a much higher place to go.
Instinctively, if I understand correctly, that both existing microfluidic systems and your system have lithography as the underlying manufacturing component. How come? And like yours has like circuits integrated, and that's so you can get like these sensors. But if the underlying creation process is the same, why is microfluidics so much more expensive than the Iku device?
So that device I was just referencing is not made with lithography. It's a molded.
Okay.
It's a molded device. But the key thing actually is that they don't have active. There's a big divide in microfluidics between passive and active microfluidics. So passive is like paper microfluidics or something, right? So your pregnancy test, it's a paper microfluidics, just kind of like does one thing, doesn't have feedback in it, doesn't really have control and regulation. And then really separate is like, okay, can you come in here? Can you sense things and change things as they're going on? And most of the systems right now do not multiplex the control aspect across a large number of things. And the sensing part of it and some of the actuation part of it, if you if you use like molded plastic, there's kind of no way to integrate sensors easily from molded plastic. Like it doesn't come out of the factory with all of these things into it. You still have to go and like add all these things together. So then you're adding in labor costs there, right? And you're adding in all that. So even if kind of like some of the end result is in certain capabilities similar, the upstream manufacturing of it, because you can't integrate everything together, really constrains your economics on it.
And so, like, even if the lithography produced microfluidics device that's potentially on the market, that alone may cost something similar to the Iku device, but all the sensors that are added on increase the cost.
Right. Let me back up here and say that lithography as a technique does have this property where it is whatever the cost kind of like doesn't scale with how complicated you make it. The big difference is in silicon, the base cost for making it is substantially higher than the base cost for making things in printed circuit boards. So in general, this is true of almost, to my knowledge, like all forms of manufacturing. As you increase, precision requirements, you increase cost, and it tends to scale like logarithmically, right? So, if you make there are two ways that you're kind of using silicon and lithography, which is either you will, like I said, make it as a mold that you then, so you're really just using the lithography as a mold, and then you'll kind of peel this casted thing off of it, or people will actually use the silicon and they make the fluidic channels in there. But the problem with silicon is it's really expensive. Like in general, we do not make disposables out of things that are made in silicon lithography. Like it's because to make something this size, probably $400 or something.
Why, like, if it seems like the big innovation here is combining lithography, or like doing lithography on the circuit board as opposed to doing it either in silicon or like via a mold, both of which seem more expensive than the route you went. Was it simply a matter of like realizing that you could do this on circuit boards and dramatically reduce your costs? Like what?
Yeah.
Like why did this not exist 10 years ago?
Right. So I think the first is that different worlds don't talk very much, and in this case, like the tool builder world and the tool user world are very distinct. And the second is that to answer the question of how did I come to it, I was in my apartment in Sao Paulo, and I'd been really digging into biofilms, and I was like, okay, like just kind of like going deep on biofilms and what's going on here. I was like, all right, so much of this is about like the concentration of these things, and like they're creating these little microenvironments and all of this. And then I was really, at the time, there was this concern about, are we going to have enough capacity for bioproduction? And what I'd seen work before is in traditional chemical synthesis, they switched to continuous flow microreactors. So, like Corning Glass that makes the glass in your iPhone, they also make chemical reactors. And the benefit of this is that you can flow things together. They react quite quickly. You can pull the heat off and things. And it's really consistent. The reason you can't use that in biology at the moment is because in order to, like, traditional chemical synthesis, you're just controlling flow rate, and the reactions happen really fast normally, right? You just mix them together and it's done. But in biology, right, like, you need sensors in order to see what's going on, the environment is really much more tightly controlled, right? There's more aspects to it. And cells themselves, right, are again like perturbing the environment around them. So that was like the lens. I was looking at like how do you bring this thing that clearly worked in chemical engineering? How do you bring that to biology? And then also thinking about like these biofilms. And so I studied mathematics. I literally wrote this down as a set of axioms. I was like, what do you need? You need to be able to hold fluids apart. You need to be able to combine them together, right? You need to integrate sensors of different modalities so that you can adapt it. It needs to be small, both for mass transfer reasons, because as you get smaller, there's more surface area around, and the limitation for many reactions is literally just like how fast can you get things from the gas phase into the liquid phase, and that's purely a function of surface area. Even actually in large reactors, when they're using bubbles, the bubbles are just creating surface area, and it's about diffusion across that. So if you go small, you get that. You go small, you also get like laminar flow, which is really, really nice because it takes problems that are in that are like normally kind of chaotic, and it linearizes them. So there's a great experiment everybody should watch on YouTube of you put a couple drops of dye into this kind of gel, and the gel has a really high viscosity. And then they kind of like stir it up this way, right?
And they go backwards.
Yeah, and they go backwards, right?
Yeah.
And the idea, it's like, well, why can you do that? You can do that because in a sense it's linear, right? Whereas in a chaotic system, you'll get to some point, and now you can't tell which path you were at before, right? So these are like things, and then you need to be able to run a lot of them, both for originally it was for throughput, but that throughput idea also translates to data parallelization. And then if you need a lot of them, you also need it to be manufacturable, right? You need to be mass manufacturable, and it needs to come down. Okay, those are like the axioms. I was like, these are the things I need. And then I literally went through every manufacturing technique that I could find. I mean, truly, everything down to like, what are they doing with 3D printed glass, at the moment? And you can just kind of like knock these out for a variety of reasons. The molded polymers don't work because you can't integrate the sensors in them quickly. 3D printing doesn't work at all. It doesn't kind of matter what the modality is, because the infrastructure isn't already there, right? So it's like, if you need to make a bunch of disposables, which is great business, always make disposables. If you need to make a bunch of disposables, then you should pick something that you don't need to have a lot of capital in order to scale it, right? So you need an existing manufacturing industry for it. And just all these came back. And then ultimately, I was like, let me just reframe it. I was like, let's just like pick one of these and like optimize for that. I was like, what's the best way to build sensors? I was like, well, I mean, like printed circuit boards are like really good. And I was like, okay, can I then build the rest of this in here? Like, can I take, like, let me just, common technique, you're right. Like, can I just select some subset of this problem, optimize for that, and then force the other ones to kind of like fit into it? And it's like, yeah, okay, sensors are good there. It's good on manufacturing. Okay, here. And then after that, went to the literature. It's like, okay, here's the one person who's kind of actually done this. Go fly to England. Go work with her. And then
the University of Bath person.
Yeah.
Okay, interesting. One person in the world has stumbled across this idea. Well, I guess like surely, if every technique seems to have like its mild drawbacks, and there wasn't a single like optimal one that you stumbled across, what is the drawback of going for printed circuit board?
Okay, well, I will tell you there's the problem that you might think, and then there's a problem you'll discover. The problem you would think is that it's a kind of like a weird thing. You have to get people to adapt to it, or also like you do have to design each of those sensor domains. Just because you pick a good sort of palette to work with, you still have to like do a bunch of work. Do all this. Those all end up actually being like not that big of a deal. The harder problem is this, which is that nobody understands it.
That's true.
Truly, like nobody understands it.
I guess, like, who are you selling these to? Like, I can imagine one customer is like academic labs, maybe. And I imagine the much bigger customer are people either preparing drugs for clinical trials or generics manufacturers. How willing are they to buy the stuff? And two, is there like a customer base I'm missing?
Yeah. So I'd say like our first customer is actually the U.S. Army.
Oh.
And that's for doing something quite different from media optimization, but still within the realm of you need to explore a larger space, and current ways of doing that are like insufficient. The broader answer here of who's the customer who feels the most pain for this are the large CDMOs. So, I've spoken to people who have worked for those places. What is the thing that they talk about every year? It's yield. That's it. They actually don't have, if we're talking about like degrees of freedom for them as a company, they don't have them, right? They don't come up with their own products. Okay. They aren't allowed to like, they aren't allowed to innovate on it once the process is set. They have extraordinary downside risk if they make a mistake. And they are in a competitive marketplace with, like the pharma companies are taking the bulk of the pharma companies are getting the value capture, right? Like they ultimately own distribution. And so they're winning there. So those features make them very desirable kind of buyers for it. But media optimization is, it both happens like within pharma companies for their sometimes pharma companies manufacture their own things, but also like the process of running dynamic cell experiments like that, dynamic cell culture like that, is pervasive. That's where I think like the largest opportunity really is all of these problems in biology, many of them ultimately just reduce to how many dynamic cell culture experiments can you run. And so this is true for like new antibiotics discovery. It's true for doing things in like organ on a chip. It's true in cancer research. Like if you actually just take the lens of what are people trying to get out of this experiment? Well, they need to be able to come in, they need to be able to, perturb things over time in this, and they need to be able to read out during it. Okay. Maybe that's too big of a lens, right? Maybe there are particular areas like our system is not going to go there. Okay. There are some edge cases where like it's not going to be compatible. But there's enough of a core there where and you see the justification for this empirically is that you already see it. Like every time that bioreactors have gotten smaller and more automated, they diffuse more into the ecosystem. It gets adopted more, and people continue to want more automation, more experiments, and cheaper on it. So,
do you view Iku as like not just a media optimization company? The hope is that whatever the final device ends up looking like, it's useful for almost anything that's an in vitro system where you're trying to screen many things across it.
Yeah. Our goal is to produce 99% of the world's dynamic biological data. And the reason that that's achievable is because we do not produce that much right now. And by increasing the throughput, by increasing the relevant modalities that we're putting in and those conditions, I think that is a totally achievable thing. That's where I started in the beginning talking about like this interface between computation and biology, and there being that mismatch. Like that interface, that layer, that's what we want to build, and that's what we want to own.
I'm curious, amongst the customers right now, like maybe the department, like the military project is its own direction for selling this to either like CDMOs, pharmas, generics manufacturers. My impression is that all of these groups, like you said, don't like variability, and so they're very hesitant to buy new technology that promises like the sky and the moon. What's the hardest part about selling to these people, and how do you reassure them that like things are going to be fine?
I certainly underestimated the importance of that aspect here. I've sold a lot of things in my life so far in very different domains, and I will say that not only in biotech, not only in pharma, but for biopharma manufacturing, the level of conservatism and scrutiny is extraordinarily high. So the like wedge or way of getting into that distribution, there are a few examples. Okay, the first one is the kind of traditional way, which would be who do the CDMOs look towards? The CDMOs are not going to adopt it until they've seen a pharma company use it. Okay, pharma companies are not going to talk to you until you have a paper published from a probably a premier lab of some sort, right? The premier lab is not going to touch anything until at least you have a white paper and some connection in order to do this. And in order to do that, you need to kind of like build the device. So it's like, how do you resolve this problem of getting to that end customer and doing it? The first is that it's like there are ways of augmenting existing instruments. So the advantage of it kind of being a standalone sensing system is that you can kind of come in as just an add-on to some. You're still going to have the same economics, but now we can offer you some more data out of that same thing, and you can. That's like a lower threshold for them, and it's not involved in the actual. Like they can just throw that part of the data away if they don't like it, right? If it's not useful, so that lowers some of it.
I guess it's like cheap enough, such that like it's not a major investment to try. Okay,
right, yeah. Right. The second is and like it's like a big. I was just rereading Jeffrey Moore's Crossing the Chasm, which have you read this? Okay, I highly recommend. If it's been on my bookshelf for 8 years, 10 years. The other day I was just like, I should reread this. And I was like, my God. And the big idea is that, for what counts as a market, like what counts as a market is not only that people are sort of like buying something repeatedly, but critically that it's a group of people who talk to each other and look at each other, right? And so you've got like the academic labs who sort of like look at each other and talk to each other. And then, the pharma companies like look at each other and, talk to each other. And then the CDMOs look at each other. But the key thing is actually like the big CDMOs, like they don't talk that much. They don't associate that much with like the little CDMOs. But those are ones actually that we can sell to and get some evidence coming in there. So there's building ancillary systems that can tack on to existing things for getting in there. And then there's the other way, which is just like be so good they can't ignore you, in a sense.
I was going to ask that. I imagine like the gold standard here is like you show one of these CDMOs like here is the cost and titer of expert produced media versus the cost and titer of expert plus Iku media.
Well, actually, the gold standard is not that we say it. The gold standard is that Eli Lilly says it.
Yeah,
right. That's because that's their customer, and those pharma companies own those CDMOs.
Yeah. I guess like has any pharma like done this and they have produced, or like even you internally have like done this like side by side comparison, and you have this very clean result to share to them, Or is it more like you're still in the phase of like you're seeing the magnitude of improvement the system gives?
It's more like it will be extremely surprising if you do not get the first of all, if you don't get the economics,
Yeah.
and then also all evidence points to being able to run more in different experiments gets you to a better answer. So you can kind of work back from
Like if you follow the trend lines, like it almost necessarily has to be the case that this is better than what's currently being used.
Yes.
Okay.
Yeah.
Is it I'm curious, has there been like any early initial deployments of this, and like there will be like a white paper coming out in the next year of like, here's what we found by like using the ecosystem?
Sure. I would say it will be much like necessarily more dull than that.
Okay.
So I would separate it actually between two things. I would say there's like the hype marketing stuff to do, and then there's the like, what does a CSO actually look at, right? And from my interaction with scientists as a breed is, first of all, they are a breed, and secondly, that they are like allergic to any hype and any kind of promotional stuff here.
Yeah.
So what they want to see, and I don't need to be creative here. What they need to see is your experiments running your device with some readout, and then you take the gold standard and you replicate that, and it needs to be at the same facility, right? It needs to be that. You need to show those two, and basically the drawing, the graph needs to be obvious enough that it's like, okay, I can see how these like correlate and they scale. They don't actually need to be perfect. That's none of them are perfect. This is true. This is true for doing scale up from the benchtop to the pilot and all of these things, right? It's really just like a series of kind of like graphs, like, okay, this thing maps onto this thing, maps onto this thing. And then the next step is actually you need that replicated at another facility. So like for pharma to adopt something, it's not even just that like you need one lab. You need it to be out of like 3 different labs who all get, because like ultimately their thing is about repeatability.
Yeah, reducing variability. Okay, yeah.
Reducing variability, but then also like repeatability. Yeah.
If someone were to hand you $100 million equity for you to like push forward the mission of Iku as much as possible, like, one, I would be curious like where you would spend the money, and 2, what are the, actually I guess connected to the first question, what are the axes of improvements that still like lie ahead of the future of the device?
Yeah. I think the first thing we would do is really build this high throughput perfusion system. I would integrate Raman sensing in it. I think that's like I think that's like a killer app. I think if you do that, it unlocks so much. But also, if you go back through the literature, people have been talking about the value and use of having a high throughput perfusion device for a quarter century. And that was before we had the machine learning or AI to also like interpret that data. That was, before the problems that we're encountering are also getting, harder to manage. So I think that's that's very clearly there. Along the way there, okay, you build like organ on a chip, high throughput. That's also like a constraint right now. It's like, one of the larger manufacturers, they're moving towards it a little bit, but they still have some trade-offs as they try to move to it. Where I think is actually really interesting, and I hadn't gone down until recently, is in droplet microfluidics. So the idea of so in some sense, what we're doing with perfusion is like, okay, let's take a benchtop bioreactor and all that control, and let's shrink that down. The droplet microfluidics is more like, let's just take a test tube and shrink it really small, right? And if you shrink, that's where it's like 10x genomics That's a form of like droplet microfluidics. It tends to be more of an integration of microfluidics with some chemistry, some chemical technique to kind of either help with signaling or help with the formations of particular types of droplets that allow kind of like membranes that you can diffuse through and things. But I think what's really underexplored are two things from the customer side or from the data side. It's higher resolution, more temporal data sets from this, right? Like again, getting back to this idea that cells are time varying and sensitive and and highly parallel in like a bunch of different ways. The ability to shrink that experimental system down that much, explore the space, but not lose the temporal element the way that it is right now for the most part. I think that's like really, really powerful. And there are a couple of techniques people have been trying to get down to it. There's a technique for like getting it down to like 7 minutes now. But it's still like there's still trade-offs. Like when I look at it, I'm like, oh, they still haven't resolved these trade-offs. So that's one aspect. I think that could be enormously valuable. And then the second thing is the droplet microfluidics right now they're really focused on the formation of the droplets and these things kind of coming through. They are not as they're not really chaining things together. And in the literature, there are all of these kind of almost like transistor parts, right? Or like little parts that people have built. And you can see like there's this dream of building like truly lab on a chip, right? And the problem is that right now, as you try to build a lab on a chip, you try to do these things, there just aren't enough of the subsystems or steps that you can kind of like link together on there. So it's like you do a set of these, and it's like, okay, we got to come out of the chip, right? And it's like, then you kind of like lose all of it. And so I think it's really only in the past 5 years, and then with our technology for being able to like actively manipulate things in there and do the feedback, I think like rather than conceiving of lab automation as automating manual tasks, which has a hard upper bound on how much efficiency and capability that you will get out of it, like let's just start doing what we did in other industries, which is be like, no, okay, we have to start over and we have to kind of like build some of these things in here. But we've already built a lot of them now. Why don't we actually start building that lab on a chip?
I remember for my lab automation article, one person remarked to me that it's a shame that liquid handlers have become so popular because like biology happens at much smaller scales than that. So like you're making a system very large when it doesn't need to be that large.
It's okay. Like I don't know if you've ever seen these like robot arms that get like a cup of coffee and then they've got them in like the San Francisco airport. Terrible idea. And it's like, okay, you go over and it's like, okay, the machine picks up the cup and then puts it over here and does the grinder and like brings it to you. Okay, what that's doing is automating a manual task. It's taking the way that humans just have kind of done something and then been like, I'm just going to throw an arm or an anthropomorphic thing on top of it and then duplicate it. Okay, and the result of that is honestly not that great, right? Like there's a reason that those things will continue to not take off in any sense of a novelty, And compare that to your Nespresso, which still has like an interface. You still need to get your cup, but like far better, right? Like the Nespresso, they're like, oh, actually, like let's integrate like the actual heater and the automatic dispenser and all of these things, right? And they made it much more compact and says, or like your coffee vending machines also works for this, right? Neither one of those are trying to just take the human steps and then just be like, literally wherever the human is, we'll just put this thing in here. And that's what I'm seeing happening right now in lab automation in general. And I don't just think it's like lazy. I do think it's lazy. I don't just think it's lazy. I think it's also like close to philosophically like a crime. I think it's a crime.
Because you think like for automation to truly be useful, like there needs to be a new way of interacting with the underlying systems.
Yeah, it's just they're not really thinking through the problem.
Well, I guess like one argument is that it's easier for these things to get adoption if you are allowing them to work in the exact same environment that humans are able to work in.
Yeah, and I think that makes sense for things like machine tenders for 3D print or for CNC machines, right? But what's the difference? Well, the CNC part is 100 millimeters, right? Okay, so it necessarily has to be closer to human scale. But look at what's happened in the most kind of like useful places for robotics and a heuristic you can use is if it says robotics in it, it's not really that useful. Whereas if it says what it just does, then it's successful. So a dishwasher is like a very useful robot, right? Self-driving car, very useful robot. And in the industrial space, it's mainly around like logistics and moving things, right? So the really successful ways of actually leveraging automation, first, they respect they respect like what the real goal is, and they respect kind of like the limits of that of the thing you're trying to manipulate. So if the things you're trying to manipulate are like grocery things, one way could be like, let's take a humanoid and it goes to the grocery store and it like picks up things off the shelf. That's what people do, and that's what these humanoid companies want to do. And the alternative would be like, actually, if you look at the logistic companies that do the best of it, it looks nothing like that at all, right? It's some like huge grid. It has these things like running around like crazy, and all they're doing is like picking up these things and setting it down. And there is no way that a humanoid system can compete with that, right? There's no way that this lazy, and you can let the economics kind of like decide that over time. I just this idea that it's like actually pushing the lab forward, I don't really buy. I just like I also like I do not see like Eli Lilly or Johnson and Johnson putting a robotic arm near their lab bench. I think what Ginkgo's doing with their lab automation system, those carts.
Yeah.
Okay, right. Like, I think that's like at least sort of like a reasonable compromise in a sense. We don't need to go and re-engineer each of these things that already exist. If we can literally just like make the interface to them easier, okay. But then I think like the real goal should be like as much as possible, if the economics fit, just like think through the problem correctly. Just put it on chip as much as possible. So
that makes sense. I think those were the last questions I had. Thank you so much for coming on.
Thank you for having me. Yeah.